And particularly when a PhD period comes to an end, you know, that becomes very difficult for a lot of students because they think, you know, I want to finish in 5-6 months' time. I cannot do another experiment. But all the way until the end of your project, when you were in the middle of writing, if we suggested another experiment, your face lit up like, oh, you know, I can do another experiment. And you always were ready to go for it, which was really nice. All right, so, but after, you know, all the niceties, of course, I have to also, you know, ask a little bit of critical questions. And I'd like to pick up on the first question of Professor Samson, because it actually corresponded a lot with mine, but I adjusted it a bit. Is the relevance in your title already of Arabidopsis as a model species? Okay. And you answered quite nicely, but one thing that actually struck me while you were at it was, what if you think about your research in the context of species that are there for a long period of time, like trees?
Yeah.
I mean, so particularly the transgenerational effect, that in Arabidopsis, I mean, it grows so short you have to do it across generations, okay? Before your experiment is done, the plant's dead. But, I mean, how would you think that sort of this transient exposure to a heat stress, I mean, how would that work in a tree? I mean, would you also have to wait till the next generation to have an effect, or I mean, could it even, could the same mechanisms go, say, from year to year in the same plant?
Yeah, this is a quite interesting question. In our experiment, we tested the transgeneration effects. For example, in trees, it's impossible. But what can be there, for example, during the growth of the of a tree, for example, this year it was a drought period, and it was a longer stress exposure. And then there was again water, and then subsequent drought again. So this training of the transcriptional responses in general can help them for the next growing season. I mean, it can stay in the cells to better cope in the next season, for example. So basically, how to say it? The stress memory of one stress exposure can be helpful for the trees in the next stress stress exposure.
And in terms of mechanisms, I mean, you're suggesting, you know, DNA, epigenetics, methylation. I mean, how does that? I mean,
how does it?
You put it very much in the context of transgeneration. Do these trees have to wait till the next generation before this methylation mechanism comes in play, or?
No. For example, trees, I mean, they have growth throughout their life cycle. For example, epigenetic mechanisms are not only transmitted through germlines, for example, through one parent to the progeny. It can be somatically propagated also. For example, in the first year, we expose plants or trees to the stress, and that stress memory can be there in those cells next time when there will be growing season, and it can be transmitted to the growing cells as well.
Yeah, I think so too. I think that, I mean, is quite a fascinating thing. Probably you might even be able to investigate that even in the short Arabidopsis by harvesting it before it actually sets seeds and see how much of the epigenetic mark. Suppose that we find, you know, it is actually there already. Sort of completely to the other end of the spectrum. I mean, resilience to stress is something that's very important for agricultural crops.
Yes.
If you can make an agricultural crop that is much more heat or drought tolerant, you know, that is worth quite a bit of money. And you know, transgenics, at least in Western Europe, doesn't seem to be a way out. You know, it's going to be more and more difficult to get that into the field. So I was wondering, you know, whether your results actually, you know, give you a potential mechanism for application. Have you thought about this? And how would you, you know, broadly think you could use these results to apply them for having, you know, better resistance in crops?
It's quite interesting. For example, as you said, I mean, transgenics, making transgenics is not always a good idea to make crop tolerance. So from here, this experiment, we showed that you can use environment as an agent to induce tolerance in the plants. For example, by doing repeated selection. So there can be various ways of doing this. For example, either, I mean, people are doing it in like various crops also, not at the transgenerational level, but at the level of one generation. They usually do the seed priming, for example, with different kinds of, for example, if there is herbicides coming in the fields, so they usually treat their seeds with the herbicides, so they have their machinery already upregulated. And then when they face those natural catastrophes, so they can grow better. So this is one thing. And the second thing could be the problem here is with these transgenerational changes is they are reversible. So we don't know, I mean, whether it will be useful all the time for a plant if there will be no stress at all. So these plants can revert back to their original state. So next year we will not have any tolerant plants. I mean, it's not like breeding where we are mixing two traits at the level of the gene.
That kind of really nicely leads into another question that I had, which is, so if you do this selection, or if you, it's also the same thing with if you did your heat stress experiment, where you get all these molecular changes, particularly upregulation of antioxidant system, it makes the plant more resistant to stress. So it makes a better plant.
Yes.
Why hasn't evolution selected for better plants, and why is there in your epigenetics, as you say, a method to actually switch it off? I mean, if the plant is better, it should be selected and should be maintained, right?
No, it could be, we have to see it in a different way. For example, if we have a continuous stress exposure, I mean, all the generations of the stress, so that trait which has been modified, for example, the upregulation of the antioxidant system, it is useful for the plants. For example, if there will be no stress exposure in any generation, that is an extra burden in terms of energy for the plant also. For example, to invest more on antioxidant than on growth. I mean, for example, and the evolutionary process, I don't think so. I mean,
does any of your data support that statement? I mean, like, are particularly these multi-generation selected plants, are they less fit under optimal conditions?
Yes. For example, from my fitness results, I think so. Yes, because stress memory is not good when you don't have stress in the plants. For example, it is only useful, which I also saw here. For example, see here. For example, these are the control, and they are lower.
Mm-hmm.
So it's
so it very nicely makes your point.
Yeah.
Okay. All right. I think with a nice strong answer is a good time for me to stop and hand the microphone over.
Thank you very much.
Okay, thank you, Eric. And